So welcome to our first of two screencasts on enzymes. This screencast looks at the structure of enzymes, how enzymes work, and the effect of various environmental factors on enzyme activity. So starting us off, remembering at GCSE you would have learned that enzymes were biological catalysts. We need to develop that idea here, and the key ideas you need to get from this slide is that enzymes are globular proteins, and we'll talk about what the word globular means in a second, and that they have a specific tertiary structure, okay, and they catalyze metabolic reactions. Okay, so dealing with some of those ideas, first of all, the idea of a globular protein. Globular means that it's got a compact three-dimensional structure or tertiary structure, usually with hydrophobic R groups on amino acids, orientating themselves into the inner part of the molecule, away from the water in the solution, and the hydrophilic groups on amino acids, tending to orientate them to the outside of the molecule to be in contact with the water which surrounds them in their solution. Okay, so that creates this compact tertiary structure with lots of tertiary folding of the protein. On the outside of the enzyme there is a region called the active site, and this is the region that binds to a molecule that we call the substrate being the molecule that the enzyme acts on. Okay, so the first ever enzyme catalyzed reaction you probably saw was starch and amylase. Okay, so amylase is the enzyme and starch we describe as a substrate. Okay, so the starch molecule will bind to a region on the amylase molecule that we call the active site. Okay, and lastly here, the word catalysis basically means speeding up a chemical reaction. Okay, we do that by reducing the activation of that chemical reaction, and enzymes, as we said, are biological catalysts. Okay, so they specifically speed up metabolic reactions. These are reactions that occur inside cells, or sometimes, as we'll see, outside cells too. Okay, so I've got an animation to illustrate the basics of enzyme activity.
Enzymes are protein molecules that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, is the area where enzyme activity takes place. An enzyme works by binding to molecules, called substrates, at its active site. The job of the enzyme is to convert the substrate into a different product or products through a series of chemical reactions. Following the reactions, the products are released and the enzyme is free to act on another substrate.
And moving on, we also have a nice little diagram here which illustrates the structure, so some of the ideas. So here's a protein molecule, okay, and it's globular. That means it's got a compact structure, highly folded. Okay, so all the secondary structures fold in on each other to form a highly folded tertiary structure. Okay, on the outside of the molecule it's got this region which we call the active site, and that's the site to which the substrate binds. Okay, and what we usually find, or we always find, is that there are various amino acid R groups, okay, which are around the active site, these red ones here for example, which will chemically interact with the substrate. Okay, so they might have ionic bonds which bind to opposite ionic bonds on the substrate, for example. Okay, so that it's not just a case of a geometrical fit, there is chemical interaction between the active site and the substrate too. This next slide looks at two different types of enzyme. We can classify enzymes as either being intracellular or extracellular. Intracellular, these enzymes are enzymes that we find inside the cell cytoplasm or somewhere within the cell, and they act on substrates in the cell and produce products in the cell. Okay, they can also be attached to membranes in the cell too. As opposed to extracellular enzymes, these are produced in cells and then released from the cell, or the word secreted from the cell, and they perform catalysis reactions on molecules outside of the cell. The key example we have is digestive enzymes, so that's starch digesting enzyme. Amylase, it would be a very good example of an extracellular enzyme produced in a cell in the salivary gland, released into the salivary duct, which then goes into the mouth where it digests starch. Okay. Other extracellular enzymes which we'll come across later in the course, which are very important, are those enzymes produced by microorganisms. A lot of microorganisms feed by secreting enzymes into their environment, digesting their environment down, and then absorbing small monomer molecules from their environment. Okay, so those would also be examples of digestive enzymes that fungi produced, bacteria produce, etc. Okay, just to show you a little diagram below to illustrate the difference between the two types of enzyme. Okay, so for example, we are dealing with extracellular enzymes first of all. Here is a cell. For example, this could be a amylase secreting cell in the salivary gland. Okay, so there would be a gene that encodes for our amylase molecule here. Okay, that's transcribed into a molecule called messenger RNA. Messenger RNA diffuses out into the cytoplasm, where it attaches to a ribosome on this rough endoplasmic reticulum. The ribosome then orders the amino acids, okay, in the right primary sequence to make an amylase molecule. Okay, that primary sequence then folds into distinctive secondary structures, which then fold in on each other to form tertiary structures. Some of that folding would occur in the Golgi body, okay, and then the Golgi body then passes the amylase molecules onto these structures here, these sort of membrane-bound packets, which are called secretory vesicles, and then those secretory vesicles fuse with the cell surface membrane, releasing the amylase into the salivary duct by the process of exocytosis. This next slide looks at all sorts of things. Okay, so at the end of this slide, you need to be familiar with the idea of specificity, active site, which we've already looked at, lock and key hypothesis, induced fit hypothesis, enzyme substrate complex, enzyme product complex, and lowering activation energy. Okay, so dealing with lowering activation energy first. Okay, enzymes, the way enzymes speed up the rate of biochemical reactions is that they reduce the activation energy of those reactions. All reactions have an activation energy. Okay, so typically this little graph here shows the energy in substrates and the energy in products. Okay, but to be able to get our reaction to occur, we have to overcome activation energy. Okay, so very simple terms, striking a match, obviously you've got the tip of your match, okay, which are the substrates, okay, and they react with oxygen in the air, okay, to produce some sort of chemical reaction, okay, but we've got to provide activation energy, which in the case of a match we do by striking, okay. So what enzymes do is that they reduce the activation energy, okay, which increases the rate at which that reaction occurs, okay, and I've got a little animation, hopefully to illustrate how enzymes, how that works with enzymes.
For any reaction to occur, even one that releases energy, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, EA. This required energy input represents a barrier that prevents even energy-releasing reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules, referred to as the enzyme's substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction. Because of the specific fit between enzyme and substrate, each enzyme can catalyze only one kind of reaction involving specific substrates. Thousands of different enzymes may be required to carry out all of a cell's metabolic processes.
And moving on to the next idea here is these are two models which try to describe how enzymes work. Remember, enzymes are tiny molecular structures which we could never see. Similarly, the substrates and the products are not discernible even with the very powerful transmission electron microscopes. We can never actually see what's going on, so we have to have some sort of way of visualizing what's going on, which we produce with a model or a hypothesis. Okay, the original model that they put forward for enzyme action was called the lock and key hypothesis, which is an analogy between a lock and a key and a substrate and an active site, suggesting the key is a bit like a substrate molecule which fits into the lock, or in this case the substrate fits into the active site to form what's it's called an enzyme substrate complex. Okay, this term here, enzyme substrate complex. Okay, you can litter your answers and exam questions about enzymes with this term here, enzyme substrate complexes. Examiners love it. Okay, and the product formed, okay, therefore doesn't fit because the product is different from the substrate. It no longer fits the lock, or the active site, and therefore it moves away or dissociates from the active site. Okay, so that explains how enzymes are specific to substrates in the same way as a lock is a specific to a key. Okay, so the idea every different substrate that we occur in a cell has to have a different enzyme to be able to catalyze a chemical reaction that involves that substrate. Okay, the limitations of this lock and key hypothesis, as whilst it very neatly describes the specificity of enzymes, it doesn't in any way suggest how catalysis actually occurs. So a more recent hypothesis that's been put forward is called the induced fit hypothesis. Okay, and that suggests that enzymes have to alter their shape, okay, alter the shape of the active site to accommodate the substrate. Okay, so it molds around the substrate. Okay, so the protein of the enzyme molds around the substrate. This results in a redistribution of electrons around bonds in the substrate, which make the reaction more likely to happen, i.e. reduces its activation energy. Okay, so this hypothesis explains the specificity, but also goes some way to explaining how catalysis actually occurs. Okay, and again I've got two little animations to illustrate those ideas. First of all, lock and key. So here I have a substrate molecule which has got a particular shape, and I've got an enzyme which has got its active site, which is complementary to that shape. Okay, so that suggests, that explains why, sorry, just pausing it there, that explains why this substrate can only fit to this enzyme. Okay, the specificity of enzymes. Enzymes will react with only one particular shape of substrate, okay, one particular substrate molecule. Whilst the induced fit model, or the induced fit hypothesis, suggests that here I've got a substrate molecule, the blue substrate molecule, binding to the enzyme, okay, but then the enzyme starts to change shape around the substrate, molds itself around the substrate, and in so doing puts strain on bonds, redistributes electrons, and the substrate in that case splits into two products which then diffuse away from the enzyme. This slide looks at the effect of pH on enzyme activity. pH on enzyme activity. pH, the idea of pH is to do with the amount of hydrogen ions in solution. It's one of those inverse scales, so the lower the pH, the higher the amount of hydrogen ions, the more acidic, the lower the pH. Okay, so what we tend to find is if we have a graph of pH against rate of reaction of an enzyme controlled reaction, we get this shaped curve here, where there is an optimum pH for which that enzyme operates, and different enzymes have different optimum pHs. Okay, but a change in pH, either a reduction in pH, okay, from that optimum, or an increase, okay, in pH from that optimum, results in a reduction in the rate of reaction. Okay, so that's a description of what the effect of pH on an enzyme-controlled reaction is. The explanation of why that occurs is that changes in pH cause hydrogen bonds and ionic bonds in the tertiary structure of the protein, which is the enzyme, to be disrupted. Okay, so the disruption of those hydrogen bonds and ionic bonds causes a change in the tertiary structure. Okay, and therefore a change in the shape of the active site, so that the substrate can no longer bind to the active site to form its enzyme substrate complex, as I've outlined here, and that results in no product forming. Okay, so that explains why there is a decline in the activity of the enzyme. Okay, the other factor is hydrogen ions can also disrupt the ionic and hydrogen bonds between substrate and active site. Okay, so there could be bonds between the substrate and the active site which could also be disrupted by these hydrogen ions. Okay, and I've also written enzymes have an optimum pH which is different for different enzymes. Okay, so for example, pepsin has an optimum pH which is probably at about 2, very low pH, because it operates in the stomach, which is lots of hydrochloric acid, whereas the enzymes that operate in the small intestine have optimum pHs of slightly above 7, okay, because it's slightly alkaline due to the alkaline bile which is released into the small intestine. Okay, so to illustrate that, okay, here I have a typical enzyme fitting into its active site. Okay, so sorry, this is the enzyme down here. This molecule here is the enzyme. This is the substrate molecule, and at the optimum pH, our enzyme fits nicely into its active site. Okay, and there are, in this case, I've got ionic bonds which are holding the substrate to the active site. Okay, now what I'm saying is that if we change the pH, okay, of our environment, the hydrogen bonds get small bits of very positive charge, protons get in amongst our tertiary structure, or our structure of our molecule, and disrupts the hydrogen bonds which hold that tertiary structure together, which changes the shape of the molecule. Okay, and not only that, but the hydrogen ions will also bind to these negative charges in the active site. Okay, and the consequence of that is that our substrate molecule can no longer bind to our active site. Okay, and therefore enzyme substrate complexes cannot form, and therefore no product can be formed. Okay, because of the change in shape of this molecule here, the enzyme, but also the attraction of these protons to the negative charges around an active site. Okay, which means that this substrate molecule can't get access to the active site. This next slide deals with the effect of temperature on enzyme activity. Okay, so when we draw a graph of the effect of temperature on enzyme activity, we notice that low temperatures there's an increase in rate of activity, okay, until a maximum is reached, which we call the optimum temperature for that enzyme to work. Okay, and increases in temperature beyond the optimum result in a dramatic decline in the rate of reaction, or the enzyme activity. Okay, so when describing the graph, always describe both phases, this phase and this phase. Also, there's usually marks on giving values from the x and y axis. Okay, so to say that between this temperature and this temperature, the activity rises from this rate to this rate, okay, whatever you read those values off the graph. And then more marks for describing between this temperature and this temperature, the rate declines from this rate to that rate. Okay, so that's a description. Now the explanation of that, explaining the first phase, the upward phase of this graph, okay, that's due to more kinetic energy. Okay, you heat your reaction mixture, both the substrate and the enzyme, okay, which are in solution, get more kinetic energy, therefore they move around more, okay, and therefore there's more likelihood of them colliding. If they collide, there's more enzyme substrate complexes forming. If more enzyme substrates forming, there are therefore more enzyme product complexes forming, and ultimately more product forming. Okay, so that explains that phase of the graph. And then at higher temperatures, what happens is that the bonds within the enzyme, okay, they start to vibrate more, okay, and that starts to disrupt bonds within the tertiary structure of the enzyme, which changes its shape, okay, changes its tertiary structure, which changes the shape of the active site, okay, which means that enzyme substrate complexes can't form, okay, and if no enzyme substrate complexes can form, we can't make enzyme product complexes, and if we can't make enzyme product complexes, we can't make product, okay, and the big word that we have to describe all of this is the word denatured, okay, but don't just use the word denatured when answering a question on this. Describe or explain what's happening in terms of the disruption of the bonds due to the vibration of atoms, which alters the tertiary structure, which alters the active site, meaning no enzyme substrate complexes forming. Okay, use all that language when you're answering questions on this. Okay, to illustrate what I mean by all of that, a little simple little thing here. Imagine I have an enzyme. Okay, here's an enzyme in a beaker of solution. Okay, obviously there's no substrate in here, so there's going to be no reaction because no enzyme substrate complexes can form. I then introduce a substrate molecule, okay, and at low temperatures the substrate molecule and the enzyme will be moving around very slowly because there's very low kinetic energy, okay, and the likelihood of a collision occurring is very small, okay, because they're moving around so slowly. Okay, I then heat the reaction up, and both the enzyme and the substrate will start to move around more, okay, and therefore there's more chance of that collision occurring, okay, and therefore enzyme substrate forming, and therefore product forming, okay. Now that occurs up to the optimum temperature, okay, when the rate is at a maximum, but at that point what happens is that the heat will cause a disruption in the hydrogen and ionic bonds that hold this tertiary structure together, altering its shape, okay, which ultimately means that the substrate, okay, although it will be moving around with a tremendous amount of energy, it will collide frequently with this structure here, but because the active site is no longer complementary in shape to the substrate, no enzyme substrate will form, and therefore no product will be formed. This slide here looks at the effect of substrate concentration on the effect of substrate concentration on enzyme activity. Okay, so if we look at a graph of increasing substrate concentration with rate of reaction, what we notice is it's a two-phase graph as well. Okay, at low substrate concentrations we get an increase in activity until a point is reached when a maximum rate is achieved. Okay, so we can't increase the activity above this maximum rate by increasing more and more substrate. Okay, so that's the description. Okay, the description of what's going on here. Okay, so substrate are just a convention we use in science. Substrate in square brackets, okay, denotes concentration of. Okay, so whenever you see that square brackets around something, we mean concentration of. So increasing the concentration of substrate results in more collisions between substrate and enzyme, okay, and therefore more enzyme substrate complexes forming, and therefore more product. Okay, so that explains the first phase. Okay, so we increase as we put more substrate in, there's more substrate molecules, and therefore more chance of collisions between substrate and enzyme. Okay, and that continues until this point here, okay, when we reach a maximum rate, okay, and that rate is achieved when all the active sites are occupied with substrate. Okay, so adding further substrate doesn't increase the amount of enzyme substrate complexes because the enzymes and their active sites are the limiting factor. Okay, there aren't any more available active sites to bind to substrate. Okay, so the rate of reaction is running at its maximum. Okay, so illustrating that below, if I were to use this little analogy, okay, so just take all these substrate molecules out first of all. If you can imagine us having my reaction vessel, my beaker, with some enzyme molecules in it, denoted by these red blobs here, these are enzymes. Okay, now when there's no substrate concentration, okay, as we have at the moment, there can be obviously no enzyme substrate complexes forming and no product forming, therefore the rate of reaction is 0. Okay, however, if I add substrate molecule in, okay, what will happen is it will eventually bind to an enzyme and form an enzyme substrate complex, the product. If I add more substrate, keep adding more and more substrate, I'm going to increase the collisions, increase the amount of product, and that will continue to happen until all of the enzymes are occupied with substrate. Okay, at that rate we have our maximum rate of reaction, okay, and at this point adding more substrate into our reaction vessel is not going to increase the amount of enzyme substrate complexes forming, and therefore the product won't be produced at any higher rate. Okay, so that's a little illustration of the effect of substrate concentration on enzyme activity. So this slide looks at the effect of enzyme concentration on enzyme activity. Okay, so a graph of enzyme concentration, increasing enzyme concentration on the x-axis, and rate of reaction or of enzyme activity on the y-axis shows a linear relationship. So increasing the enzyme concentration increases the rate of reaction. Okay, so again, the way to explain what's happening there, enzyme concentration, again square brackets denoting concentration of, so concentration of enzyme results in more collisions, okay, therefore more enzyme substrate complexes forming, okay, therefore more product formed, okay. If the substrate was limiting, so if we actually had a limited amount of substrate, okay, we would eventually achieve a maximum rate here, okay, like we did in the last slide, okay, with increasing substrate concentration, because at some point the substrate would become limiting, okay, but when the substrate is in excess, okay, so we've got lots and lots of substrate, okay, what happens is that the relationship is linear here, okay, so just illustrating that below. Okay, so this time I've got my substrate molecules, okay, here's my blue substrate molecules in my reaction vessel, my beaker, whatever it is, and at the moment there's no enzyme in there, so no rate of reaction because no enzyme substrate complexes are forming. Okay, if I add enzyme, okay, that will move around, eventually collisions will occur, enzyme substrate complexes form, product forms, okay, adding more enzyme, more collisions, more enzyme substrate complexes, therefore more product, okay, so we get a higher rate of reaction, and we can keep doing that, okay, we keep getting more and more enzyme substrate complexes, more and more product, okay, and that will continue, okay, as long as the substrate's in excess, okay, we've got enough substrate, but when we reach a situation where all of the substrate is bound to enzymes, okay, adding further enzymes, okay, doesn't result in the production of any more enzyme substrate complexes, okay, so we do reach that maximum rate eventually when we add more and more enzyme. This slide looks at experimental considerations when doing experiments on all the things we've just looked at, so effects of pH, temperature, enzyme concentration and substrate concentration on enzyme activity, things we ought to think about if we're doing experiments. Okay, the first thing to consider is that the three types of variable which you'll have come across before. Okay, if I look down here, when we draw a graph of an enzyme controlled reaction, we've got the independent variable that we put on the x-axis. Okay, so we could be affecting the change of pH on enzyme activity. So in this case, the independent variable would be the pH. Okay, and we sometimes call that the input variable, the thing that the experimenter is deliberately manipulating. Okay, that's the independent variable. And then the outcome, okay, what changes, in this case the rate of reaction, is called the dependent variable, which we always put on the y-axis. Okay, you also need to, because as we know, lots of things control or alter the rate of an enzyme controlled reaction, not just pH. Okay, so anything else that might influence the rate of reaction we have to control or standardize between experiments. Okay, so if you had, let's say, different tubes of different pHs with enzyme in it, okay, you would have to make sure that the amount of substrate was the same, that the temperature was the same, and the amount of enzyme was the same, because all of those would also affect the rate of reaction. Okay, and those are all what we call controlled variables. Okay, so the three variables: independent, dependent, and controlled. So think about those. Okay, the other issue when dealing with enzymes is obviously enzymes are usually colourless, invisible molecules in a colourless, invisible solution reacting on colourless, invisible substrates, producing colourless, invisible products. Okay, which makes it very difficult to actually work out what is going on and how fast it is going on. Okay, so to be able to work out the rate of reaction, okay, enzymes which cause a change of state, okay, from substrate to product are very useful, and the enzyme catalase does exactly that. Catalase takes a liquid hydrogen peroxide and turns it into the gas oxygen. Okay, so we've got a change of state from liquid to gas. Okay, and this gives us an easy way of measuring the amount of product, oxygen in this case. Okay, so we will do this, and you'll have done this in the lab yourself. Okay, so we could have a situation where we have our hydrogen peroxide here, okay, and then we can add some catalase reaction here, and then it starts to produce some gas oxygen, and we can measure that oxygen. So this could be an inverted measuring cylinder where we have a scale on the side, so we can measure the amount of oxygen in a given time. Okay, and we put different amounts of catalase in, okay, and we would get different amounts of oxygen produced, or we could alter the temperature of this. Okay, we could do all sorts of things. We could do all of our reactions, effective substrate concentration, effective enzyme concentration, effective pH, effective temperature. We could do all of these experiments by manipulating the environment inside here, and we could measure the outcome by the amount of gas produced in a particular given time to give us an indication of the rate of reaction. Okay. Other ways of, we could also sometimes, the reaction lends itself to measuring the amount of substrate, or how quickly substrate disappears. Okay, starch disappearance with iodine, with amylase is a good one. Okay, so this arrangement down here, where what we would do is we would have our solution. Okay, here's our amylase and our starch. Okay, we put that into our reaction vessel here, we mix them together. Okay, so the amylase starts to break down the starch. Okay, and what we do is we have these spotting tiles which we put spots of iodine in. Okay, and what happens is that at certain time intervals we take a sample out of our reaction mixture and spot it into a tile. Okay, so obviously to start with we still have starch left over, but a point is reached when the starch doesn't turn blue-black. Okay, so that tells us when the reaction is complete. Okay, so there we're looking for the disappearance of the substrate, the disappearance of starch. So we're looking for the time when iodine no longer gives a positive test for starch. Okay. Or sometimes you can measure the the product, the product, so we could, for example, rather than measure the disappearance of starch, what we could do is we could take small samples out of our reaction mixture and start to measure the appearance of our product, which in this case would have been a reducing sugar. So we could have done a Benedict's test on products, okay? Or sometimes, there's another nice example here, lipase, which breaks down fats or triglycerides into fatty acids and glycerol. The fatty acids reduces the pH of the solution, and we can monitor the pH of the solution with a pH meter or some universal indicator paper or whatever. Okay, so we can get an indication of how quickly the fatty acids are being released by the lipase. Okay, so those are all ways in which you can investigate the effect of pH, temperature, enzyme concentration, substrate concentration on the rate of reaction, which is controlled by enzymes.